Today I'll be talking about how adrenaline, also known as epinephrine, may affect the body's heart rate, which is an example of a G-protein coupled signaling pathway. This pathway consists of adrenaline molecules being accepted to a receptor, which in turn activates G-protein and performs a series of amplifications in order to increase glycogen phosphorylation, allowing more glucose to be hydrolyzed and thus more energy to be harnessed for supplying an increase in heart rate. G-protein coupled receptors are extracellular receptors that regulate metabolic activities of the cell. Here is the overall process of the G-protein coupled signaling pathway. The G-protein consists of three parts: alpha, beta, and gamma. While the receptor is inactive, the alpha component is bound with a GDP. First, a hydrophilic ligand binds to the transmembrane receptor protein. The protein's shape is subsequently altered, which then affects the G protein connected with it. The alpha component is stressed and released the GDP, replacing it with a GTP, then is detached from the G protein. Finally, the detached alpha component binds and activates the target protein, triggering a series of cellular responses. When adrenaline binds to the receptor as a ligand, the target protein of the GTP-bound alpha component is a transmembrane protein called adenylyl cyclase. Adenylyl cyclase is an enzyme that converts ATP to cyclic AMP when active. CAMP, which is a secondary messenger, in succession binds to protein kinase A. This activates the pKa, which causes a catalytic subunit to be released from its regulatory subunit. This subunit then binds to and activates phosphorylase kinase, which in turn activates glycogen phosphorylase, an enzyme that phosphorylates glucose monomers to separate them as glucose 1-phosphate from glycogens. Ultimately, the increase in glucose concentration allows glycolysis and subsequent catabolic reactions to produce more ATPs. Each of the messengers here may activate multiple proteins of the subsequent layer, which allows a small amount of adrenaline molecules to amplify into a greater cellular response. The process terminates by adrenaline molecules detaching from the receptors, which reverts GTP to GDP bound to G protein, hence stop the production of CAMP and concludes the amplification operation of all layers. In this process, adrenaline molecules are endogenous agonists, which means they are ligands that are produced inside the body and activates a response. They are endogenous as they are hormones produced by the adrenal glands, a part of the kidneys, and circulates to cells through endocrine signaling. The specific effect they have on the G protein signaling pathway is changing the shape of the receptor, and thus causing a series of secondary messengers to activate the next layers of proteins and amplifying the response, hydrolyzing glucoses from glycogens. This allows increased cell work rate, and the extensive results they have on the entire body includes the increased heart rate and muscle strength.